on. Welcome, everyone. Thank you for taking time to join this discussion about linking habitat to stand structure. Okay. Imagine you live in rural Bulkley Valley and you look out your window and you see a mature aspen forest. Maybe picture something like this stand. It's easy to be lulled into thinking that your backyard will look like this for your lifetime and that it's always been that way. It's just the way it is. But if you think about it, perhaps 80 years ago, it was a sea of black snags with a brilliant carpet of fireweed and vigorous bright green aspen seedlings popping up. Perhaps not visible yet are small subalpine fir and pine and spruce seedlings. Also intermixed might be some willow and some high bush cranberry shrubs that are also resprouting. And we know in another 80 years, this forest will likely have transitioned to a darker conifer forest where the spruce and the pine outcompete the aspen, whereas the aspen die. And there's few of the shrubs that the moose might like to eat in the understory. As you've just pictured, forest ecosystems are dynamic. They change over time. And those changes are driven by changing environmental conditions, by competition for resources, by successional processes, by disturbances such as fire or mountain pine beetle. We have models that allow us to predict how the trees grow, how they regenerate, how they compete, how they die over time. These models tell us about the structure of the stand and the trees and the tree species that you find in the stand. But how do other features in a forest change as the trees are changing and developing, as the stand structure changes? How do different ecosystem components, such as the shrubs that moose like to eat in this forest, or perhaps the lichen that caribou rely on in other forests, how do they also change? Where do we find these features? How long do the conditions last that support them? Will a forest always provide great lichen habitat? Or is it only for a period of time before the forest canopy changes and moss outcompetes lichen? As more of our landscape is impacted by forest management and also natural disturbances, we need to understand how other ecosystem features change and those features that are critical for other values on the landscape. So for example, in order to better manage caribou and moose habitat, we need to understand how the critical components of their habitat change as the forests are impacted by forest harvesting or mountain pine beetle or fire or any of the numerous other disturbances that might impact the landscape. When we gain this understanding of how the ecosystem components change as the stands change, oops, somebody might need to be muted. When we gain this understanding of how are the habitat features linked to the stand structure, then we can manage forests better to maintain habitat so that we can maintain all aspects of the forest that we value in addition to the trees. So how do we do this? We have forest simulation models that are focused on trees. So we have numerous models that are pretty good at predicting how trees change over time. They're primarily tree focused. They do tell us about how light in a stand changes. Some are adapted or have been developed to tell us about how carbon dynamics change. In the Northwest here, we have SOAR-TND, which has been developed to model complex stands and intimate mixtures of species. So they describe, and this little figure here shows an example of a model, that is modeling a gap cut scenario. So as an example of a complex stand that can be modeled. But how do ecosystem components change over time? I've already talked about some of those components. Sorry, I just lost my train of thought. So yeah, so we can predict how the trees grow and change over time, but how do we predict how the ecosystem components also change and develop over time? We can use linker functions. So these linker functions, so what are linker functions? They are mathematical models of our understanding of how ecosystem components are linked to different stand features. So if we know how the stand changes, then if we have a linker function that will predict under the stand type what ecosystem features are associated with that stand type. And as that stand type changes, what does that mean for those ecosystem features? So before I dive further into the linker functions, I'll provide a bit of background on the Tweeds-Marentiaco caribou herd and their winter range. That's the focus of this study. I'll talk about the habitat features that we're focused on modeling in this project. I'll provide a quick introduction to sortie and then move on to discussing the linker functions, the steps that we followed to develop them, the functions themselves, and then talk about the next steps of this project. So the Tweeds-Marentiaco caribou herd, their full range is here in west central British Columbia, circled by, it's the center of this pink circle here. You can see the Telkwa herd is at the top, just north of the Tweeds-Marentiaco caribou. And this slide is basically here to give you a visual picture of their ideal winter range. So in the winters, they migrate to low elevation pine forests that are generally poor sites that have extensive terrestrial lichen cover. Terrestrial meaning it grows on the ground. And this we'll talk about what species of lichen there are later. But this one photo is what the winter range looks like in the summer, but as you can see, it's covered in snow, but it's not covered in a lot of snow. So they focus on areas with a low snowpack so that they can dig through the snow to the lichen throughout the winter. Occasionally, depending on the winter and depending on who knows what factors, they'll move up to alpine areas where there's windswept slopes that they can also access terrestrial lichen on. So just a little bit, another photo or map to orient everybody to the location of the study. The orange circle here is the Tweeds-Marentiaco herd's full range. So in the summer, they migrate west into the mountains for calving and also for the just richer, better feeding sites. And in the winter, they migrate back east to their winter range, which is in the eastern portion of this orange circle, or it's encompassed by the green circle. And as you can see, the SBS MC2, so that's an ecological unit of similar forest types. It occurs throughout their winter range and also extends all the way to Smithers. So this is a bit of a busy figure, and we'll talk more about my field plots later, but it does show the locations of my field plots. So I spent a fair bit of time, I spent as much time as I could sampling within the winter range of the Tweeds-Marentiaco caribou themselves. However, there were numerous access issues. The barge wasn't running that summer, and there was a washout that didn't allow me to access most of this area. So we sampled as much as we could, and then we moved back to Smithers and sampled areas that were more efficient for sampling. So why the Tweeds-Marentiaco caribou? Why study their winter range? Their winter range has been significantly impacted by disturbance. It is very close to one of the centers where the mountain pine beetle epidemic in the early 2000s started. And as you can see in the photo in the center, there's extensive mortality caused by a mountain pine beetle across their winter range. There have also been significant fires throughout their winter range, and sorry, I have notes, but the Cheslasley fire burned 133,000 hectares in 2014, and the Tweedsmuir complex burned over 300,000 hectares in 2018. And there are other fires earlier in the 2000s in their habitat as well. So we don't know We know the trajectory of caribou habitat following fire reasonably well. That's been studied across Canada and across the north. But what happens to the terrestrial lichen following mountain pine beetle? We don't know as much about that. And in addition, there's salvage harvesting that's proposed and ongoing in the area, and numerous silviculture treatments are also being proposed. And there's questions about what does that mean for caribou habitat? And what does it mean for not only caribou habitat in the short term, but also in the mid to long term? And what are the best treatments that we could apply now that would maintain and enhance caribou habitat in that mid to long term? So those are questions that were being raised, and those questions precipitated this study. Okay, habitat, just a quick slide on habitat. Habitat seems like you should be able to come up with a simple, quick, concise definition of a habitat. Perhaps you can, but when you start to think about it, habitat is quite complex, especially for a species like caribou. So there's many scales you can look at habitat from. The first order being, what's the range of the species? But for caribou, they're a circumpolar species. They exist across the north, across the world. So their range is very extensive. And as you can imagine, their habitats are significantly varied as well. At a slightly smaller scale, there's a second order of habitat, which encompasses the home range of an individual animal or social group. So for the Tweedsmuir and Tiaco caribou, that's a broad range of at least 200 kilometers east to west, and extends from low elevation pine forests up to rich, wet mountain or alpine ecosystems. So it's still a very broad range of habitat types. The third order, so in this narrower scale, habitat describes the components of the habitat that you would find within the home range, perhaps, and even more specifically could be feeding locations within specific forest types. So for example, the dry pine forests. So that's kind of the scale that we're looking at for this study. The fourth order would be looking, and we are considering the fourth order as well, where you actually, within those pine forests, what do the caribou actually eat? What do the moose actually eat? So now that we have an idea of what scale of habitat features are we focusing on with this study, what are the actual habitat features we've chosen to focus on? So for caribou, primary aspect of their winter range is the food that's available there. And for these caribou, it's the terrestrial lichens, Cladina, Cladonia, Stereocolon species predominantly. I didn't find a lot of Citraria on the landscape, but they do eat Citraria as well. And then a key aspect of caribou habitat is the fact that it's not habitat itself, but so caribou's primary predation avoidance strategy is to space themselves away from wolves. And generally, where you find higher population densities of moose and deer, you're likely to find more wolves. So caribou generally separate themselves away from good habitat that supports moose and deer. So that's why you find them in these really poor pine forests where you don't find a lot of other critters. So that's a key aspect of their winter range, is that there isn't moose habitat and there isn't deer habitat. So I'm also studying, so a component of this study is to study the browse species that do make good moose winter range. And those browse species include woody shrubs, and subalpine fir and aspen trees, as well as black cottonwood trees. The shrubs that we've focused on are willow, red osier dogwood, Saskatoon, highbush cranberry, mountain ash of both species, Douglas maple, and red elderberry. Another important aspect for moose winter range is the presence of mature conifer cover, and that's important for moose for snow interception so that there's lower snowpack and for ease of travel, as well as for thermal protection. And you might think thermal protection is protection from the cold, but actually for moose, it's more important that they have protection from higher winter temperatures. And it's been shown that above -5 degrees Celsius, moose can start to overheat. So we can describe areas of high quality habitat. We can picture them in our minds. But when you start to ask, when do those stand conditions actually occur? How long after a fire or how long after mountain pine beetle do they develop? What are the initial stand characteristics that lead to this high quality habitat? So for instance, for caribou winter range, we know they like mature pine forests to have this abundant cover of lichen. But what is the density of the pine when it started that develops into this stand type? Was it an open stand? Was it a really dense pine stand? And then how do these features change as stands develop with succession is another question. So we can go into a stand and we can assess the quality now, but how will it change as the stands develop? So that leads us to sortie. So we have a model, as I talked about before. It's an individual tree model, it's spatially explicit, and it models forest dynamics. So what does that mean, really? All these fancy little yellow or colored circles here represent a stand in sortie. So sortie knows the exact location and it tracks the exact location of each tree, and it knows the species and it knows the neighborhood around each tree, and it grows the tree based on its light environment and based on the competitive environment around it. So how many trees surround it, how close they are they taller, are they smaller than that tree? Those are important features or important drivers for how the trees are modeled and growth is modeled. It also models snag dynamics. So if we can model trees being killed by mountain pine beetle, and we can model the snags and how they change over time. So if a tree dies, it doesn't just disappear, it stays in the stand and it slowly breaks down, and we know that affects the light environment around of the neighboring trees. So this figure just shows there's numerous different output you can get because it's tracking both all information about the individual trees, and then it also outputs light information. So this gray square on the bottom left just shows how the light environment might differ across the stand depending on the density of trees in each location. So how do we model ecosystem features? That's the big question. We know, as I said, we know how to model trees. We have sortie, does a good job of modeling trees and the stand structure. And this little add-on to the figures just to remind us that it doesn't just model at one point in time, or you don't just ask, what does the stand look like in 80 years? It outputs information every year along the way, so you can see how the stand changes over time, as well as what it ends up looking at like in whatever time period you're interested in. And then we know that we want to know how do lichen and the moose suitability of a site change following disturbance. But how do we get there? So for the purposes of this study, we chose to use linker functions. So we developed preliminary linker functions based on a literature review and meetings with various subject experts. And we developed these functions or mathematical models that tell us the suitability of a site to support either lichen or moose browse based on stand structure features or data. So based on the data we can get from sortie. We then conducted a field study, as I mentioned, to refine those linker functions so that they're based on field data. And then what do linker functions do? So this figure I tried to show that you take the stand structure data You then apply linker functions to that stand structure data, and you're then able to project the lichen and moose suitability. So I just have a couple slides to describe the field study that I undertook. So we selected, there was response variables were the 2 forage communities of interest, so terrestrial lichen abundance and the abundance of moose browse shrub species. The predictor variables that I was measuring were stand structure variable and numerous stand structure variables, light, site conditions, the stand age, and the vegetation community. And then I sampled across a range of gradients. So I sampled across stands from young stands to the oldest stands I could find. I sampled various tree species compositions or stands with different mixes of trees. So I wanted especially stands with few broadleafs in them, few aspen, to stands that were mostly aspen, and pine stands, as well as a mix of pine and spruce and subalpine fir. And then I sampled across edaphic gradients, so that means from dry to moist sites and from poor to rich sites. Here's a figure of the distribution of the 76 plots that I sampled across stand age, so from about 10-year-old stands up to 230-year-old stands, and from the driest stands at the bottom of the figure to the moist, rich, or moist stands at the top of the figure. So what did my plots look like? I basically followed the Beck protocol, the biogeoclimatic ecosystem classification plot protocol, but my plots are half the size. So instead of a square 30 by 30 meter plot, my large plot was 7.98 meters in radius. And within that plot, I measured trees over 12.5 centimeters, so the large trees, vegetation cover. I searched for moose browse on all the shrubs, and we counted piles of moose droppings. And we measured the height of the average canopy height and measured in cord trees to get the stand age. Divided the plot into quadrats, and within each quadrat I estimated the percent cover of lichen as well as any other competing ground cover. In the small plot, so 3.99 meter plot, I measured trees, the small trees that are less than 12.5 centimeters in diameter. Then I took 5 hemispherical photos that I used to estimate the light level within the stand. And I dug a soil pit and wrote up a site description and estimated the soil moisture and the soil nutrient regime of the stand. So linker functions, the lichen linker functions, we'll start with those. So my preliminary linker functions, which you'll remember are based on just literature review and expert knowledge. The variables that we selected that we considered most important to drive whether you would find terrestrial lichen in the stand, specifically the lichen that caribou eat, our time. So we hypothesized that it takes time for lichen to seed in, especially following fire. Competing vegetation, so the presence of competing vegetation drives the presence of lichen. Basically, lichen's strategy is to live where other vegetation can't. So they're very slow growing and they're survivors. They can survive on very poor sites. They can survive on sites with very low moisture where other species can't. And then the presence of broadleafs also determines whether a site will support lichen, because the lichen litter can just physically smother, the broadleaf litter physically smothers the lichen. So as you have higher broadleaf content in a stand, you're less likely to find lichen because they just can't, they grow so slowly they can't get above the broadleaf litter. A caveat of developing these linker functions is that they have to be based on data that I can output from sortie, from the model. So they have to be stand metrics, and sortie doesn't model competing vegetation. So instead of modeling competing vegetation, we hypothesized that light was we had to go back to basics and determine what drives the presence of competing vegetation. And that would be light. If you have more light, you're likely to have more vegetation in the stand. If you have more moisture, you're likely to have more vegetation, and as stands become richer, you're also likely to have more vegetation. So those were our preliminary linker functions. I then did my field study, as I mentioned, and we analyzed the data using likelihood analysis, which allowed us to refine which variables and which model forms best predict the abundance of lichen. And we used the data to estimate the parameters that define the best model fit, which is a lot of statistical talk for some of you folks, I'm sure, but we'll look at some pictures that describe what I'm talking about shortly. So following the field analysis, we refined the linker function down to the following 5 variables in a combination this way. So we said time was determined to be an important predictor of lichen, so it does take time for them to seed in. You need to model that. Light levels do drive whether you find lichen in a stand. Soil moisture and soil nutrient regime are important. They're considered additive instead of multiplicative, so that means, and this is somewhat interesting, so soil moisture is an important variable, soil nutrients are an important variable, but they're not exclusive. Like, you don't need to know both of them. They're additive, so they're complementary to each other. So if it's a drier site but it's richer, it's still better for lichen. If it's a poor site and it's wetter, it's still okay for lichen at its best if it's both dry and poor. And then broadleafs. And and it's also important to remember before we start looking at figures that we're not predicting the abundance of lichen, we're predicting the suitability of a stand. So it's scaled from 0 to 1. So 1 meaning if the stand suitability is 1, it's the best possible stand for lichen. It's not necessarily that you won't find 100% lichen cover. It won't be, yeah, it won't be 100%. Oops, what happened? Okay, so these are figures that illustrate 3 of the linker functions. So the first is stand age. Second is, can you see my mouse if I do this, or can you only see it if it's over here? Sorry.
No, we can see it a bit on both.
On both, so you can see it when I'm pointing to this slide?
No.
No, okay. Sorry, so I'll be looking at a separate screen. Sorry, everybody.
We can see right now on the lichen suitability graph in the middle there. Yeah.
Okay, perfect. So the first figure shows the relationship of stand age to lichen suitability. So as I said, you can see it takes time for lichen to seed in, and then once you hit about 50 years of age, you have the highest possibility of finding lichen based just strictly on age. The black dots represent my field data. So you can see the actual relative abundance of lichen across stand age. And you can see that at 100 years, you do have some of the best conditions for lichen. You also need to remember when you're looking at these figures, this is the best conditions for lichen based on that variable, so stand age. But if say you're at a 100-year-old stand, but light levels are very low, then those interact together to reduce your suitability of your stand to lichen. So you can see here in this figure, the middle figure shows the relationship with light levels. And just as I said, if you have lower light levels, which is on the left of the figure, you're less likely, it's very poor conditions for lichen. And lichen they're not plants, but they also rely on photosynthesis to provide energy and to grow, so they also need light. So this isn't surprising. And then, as we talked about before, the percent deciduous basal area also impacts lichen abundance. So as you increase in your percentage of deciduous, you drastically reduce your probability of finding lichen on that site. So this figure illustrates the effect of the last two components of the linker functions, so the effect of site moisture and site nutrients. And as I talked about earlier, so the bottom, this figure matches the Beck edotopic grid for those that are familiar with the edotopic grid, but for those that aren't, on the x-axis, the horizontal axis is soil nutrients. So on the left are the poorest sites, and on the right are the richest sites. And then on the y-axis here is soil moisture. So at the top of the figure are the driest sites, and at the bottom of the figure are the wettest sites. So you can see as we talked, as I described at the beginning, the driest and poorest sites support the most lichen. And the size of the dots here represents the amount of lichen that was found in at those points on the edotopic grid. So those plots that were the driest and the poorest plots had the most lichen. It is interesting, you can see there was a plot that had a fair amount of lichen in a richer, wetter site. But overall, this is the trend that you would see. So the red indicates the most suitable, the best conditions for lichen to be found. So the linker functions for moose. So the preliminary linker functions we hypothesized were that moose, to be a great stand for moose, is a function of there being browse available, so food available, just like for caribou, but also that you need to have a certain amount of conifer cover in the area as well. So for browse species, as we talked about earlier, the moose eat shrubs as well as broadleaf seedlings. So seedlings of aspen and cottonwood less than 3 meters in height, and then also subalpine fir that's less than 3 meters in height. Thinking about how high, generally 3 meters is the cutoff that's used to indicate if it's available for moose. If it's too tall, obviously they can't reach it. And it's also generally, it has to be above 1/2 meter in height to reflect what the browse that's available above the snow. And then, as we talked about before, similarly, we hypothesized that light, moisture, and nutrients are the important drivers or determinants of whether you'll find shrub species in a stand. So again, I did a likelihood analysis again, or I used a likelihood analysis to estimate the drivers or the linker function for the browse shrub species. I didn't use it to estimate broadleaf cover, subalpine fir cover, or conifer cover, because I can get that data directly from sortie. It predicts how the aspen component, it'll predict the subalpine fir component, it'll tell me the height of the trees. So I can just select for every stand, I can query how many trees are there under those height cutoffs, or above a certain height cutoff in the case of conifer cover. So we refined the linker functions with the field data, again, as we mentioned, and they shifted a little bit. So for shrub species, we found that light was an important driver. Actually, that should not be in there. We found that light actually isn't that important a driver. It doesn't tell us very much. If we know the conifer basal area of the stand, and the conifer basal area being the basal area of large conifer trees, that tells us more than light level in the stand, which is interesting because so the mechanism that we were we'll talk about that when we get to the figure. But so we have moisture, nutrients, time, and conifer basal area. And again, it's a suitability index on a scale of 0 to 1. So it's not predicting absolute abundance of browse species. So the edaphic figure first. So how does moose browse change depending on the moisture and nutrients of your site? As you can see, it's the reverse of the lichen linker function in general. So the wetter, richer sites are more likely to support moose browse species. but it's not as strong a relationship. And you can see there's definitely moose browse species that are found on the drier and poorer sites. And there are quite a few, in fact. And I've found, especially willow and Saskatoon, you could find them across sites, really. And you almost always, when you went to a site and you found it was a great lichen site, there'd always be the odd willow kicking around. So I found that interesting. Time is an important predictor. So as with lichen, it takes time for the shrub species to develop. It takes time for them to recover and resprout and then develop to a certain size. And then, as many folks know, as stands age, the moose browse species generally fall out. And perhaps that's related to senescence and just the shrubs getting older and just not being able to regenerate under a mature canopy. It could also be related to the light levels, but as I mentioned, we found that light level wasn't as important a predictor. So when you're thinking about stand age, it is really important to think it has nothing to do with any other aspect of the stand changing over time. It just has to do with time itself. How does that affect the presence of moose browse? So conifer basal area, we found that it seems as though there's almost a threshold, but when we're developing linker functions, we have to think about what is the mechanism and hypothesize what the mechanism is, and then whether it's an important driver for moose browse. So here we were thinking you don't find moose browse species in dense conifer stands, and especially the older dense conifer stands. So does that determine, is that an important predictor of moose browse? And we found from this, as you can see from the analysis, that it is an important predictor. So in stands that have low conifer basal area, so those could be young stands, they could be young plantations, then you have a high likelihood of finding moose browse. But they could also be mature stands that are just mostly aspen, or mostly, well, we didn't sample a lot of cottonwood, but you're more likely to find large stands of aspen. Anyway, in those stands, you also have a high likelihood of finding browse species. And then as the component of conifers increases, and the component of large conifers specifically, you get a significant decline in the suitability of the site for moose browse. Okay, so these figures, you're used to looking at the black dots in my field data, and these black dots do, but you might be wondering, why are they at 0? So that is because we don't know the moose suitability of a site. I don't have collar data that I'm using to assess whether the site is suitable for moose, whether they use it based on the density of subalpine fir. Unfortunately, it'd be great to have that information. It'd be great to inform these curves, but instead, because we don't have that information, we just had to come up with the shape of the curve based on literature and consultation with moose experts. And then I wanted, so for example, for the density of subalpine fir, this figure shows that as you increase in subalpine fir saplings in the understory, the stand becomes better and better for moose. But there's probably a maximum, and we're hypothesizing there is, that above a certain amount of subalpine fir, it doesn't matter if you have 10,000 or 100,000 stems of subalpine fir, it's just good for moose, and there's only so much they can eat. I suppose at some point it actually becomes too dense and they wouldn't be able to move in there, but we're not looking at that. So I plotted my data just so that I could see that the numbers roughly make sense, that you do find stands that have more than 3,000 stems per hectare of balsam, or say more than 5,000 stems per hectare of aspen. And then similarly with the conifer basal area, so this is, again, remember, it's the conifer basal area of trees over 12 meters in height is important because that represents mature trees that provide thermal cover as well as snow interception for moose. What are we doing for time? Oh, this is good. Okay, so this is a timeline just showing the timeline of my studies. So we developed linker functions for lichen suitability and moose habitat. The next steps are to model silviculture scenarios. So I'll be using sortie to model clear cutting, partial cutting, using small gaps, and also uniform removal, and then no harvest scenarios, and also modeling what happens to lichen and moose browse following mountain pine beetle. Once I have those model results, then I'll apply the linker functions to the output to predict how the lichen and moose suitability changes over time. So this is a figure. It's very preliminary. It's from the early linker functions before I did my field data, but I wanted to put it here to show how you could compare different scenarios. So here I had 3 different densities of pine plantations, and the figure shows that it's increases in suitability for lichen as the lichen seeds in, and as the pine stand is fairly open because there's lots of small trees there. But as the canopy closes, the suitability for lichen decreases dramatically. And then depending on the density of the stand, the suitability for lichen increases at different points in time. So that's what we're hoping to get similar figures from in short order. So in the next month or so, I'm doing my silviculture scenario modeling, and we should have more results, so stay tuned. Okay, so my take-home messages. This is my last slide. We need to understand the connections between forest structure and habitat attributes. Understanding these connections is key for managing for habitat features beyond trees. And linker functions are one tool that we can use to do that, because we do have this existing tool. We have sortie that allows us to model a broad suite of complex stand conditions and numerous different silviculture scenarios. If we develop linker functions that link the stand attributes to whether you'll find habitat features of interest, then you can predict how those habitat features of interest also change over time. So as I mentioned, up next, I'll be modeling the dynamics of caribou habitat following mountain pine beetle and silviculture treatments, including partial cutting and clear cutting. And I'm very interested in finding out, seeing what we can learn and how that can inform best management on the landscape for Tweed-Smear and Tiaku caribou. And I'm sure that's what most of you are interested, or a lot of you are interested in as well. So I'm sorry I don't have any results at this point in time, but they'll be coming soon. I'd just like to acknowledge Dr. Che Elkin, who's my supervisor, who's been amazing at supporting this work. And also Erica Lillies has been excellent. She's also on my committee, and she's been key for helping, especially with my analysis, but helping set up the field study and just general support throughout this project. Dr. Chris Johnson is also on my committee. He's a great resource to have in the world of caribou and lichen and moose. Additional contributors include Dr. Alana Clausen, who's doing her postdoc working with sortie. So that's been excellent to work with her. She's been adapting it to be able to model across different site conditions. And then Debbie Chakowski, Anne-Marie Roberts, Dr. Jocelyn Campbell, Dr. Dave Coates, Dexter Hodder, Shannon Crawley. Dexter and Shannon are with the John Prince Research Forest as well as UMBC. And Dr. Darwin Coxon. They've all been very helpful, and were some of the experts, well, they were the experts that I relied upon to inform the linker functions early on, and Anne-Marie Roberts has also been great at supporting this project throughout the project. Just wanted to acknowledge my funding sources, which is primarily the BC Caribou Recovery Program and also the Province of BC Pacific Leaders Program. So that's my presentation. I would love to invite questions and discussions, and if you have any later, please feel free to contact me. Yes, we're still in the process of doing this work, so any input, feedback, suggestions, ideas, I'm all ears.
Great. Thanks, Erin. It's fabulous.